right, I think we have recording in place. Did you all get the message? Okay. All right, so today we will have Ken Olivier presenting, and the topic for today is it safe? Toxicology and therapeutic drug development. Can you hear me okay? Okay, great.
Yes.
Okay, so Ken is going to talk about toxicology, and he's given a couple sentences here about talking through the physicochemical properties of a test article. And I don't know if he's going to actually talk about all of this, but this is toxicology and the biology of the intended target. But he's actually been developing drugs for over 18 years and is currently serving as head of toxicology and various drug development activity at Orange Grove Bio, vice president of development at TScan Therapeutics, and director of non-clinical development at Boston Immune Technologies and Therapeutics, as well as president and sole proprietor of Olivier KOnsulting with a K since inception in 2015. So Dr. Olivier's work experience is really in the biotechnology and pharmaceutical companies, spending time at numerous companies that I'm sure we'll all recognize: Regeneron, Biogen, GlaxoSmithKline, Merrimack Pharmaceuticals, Acceleron, Torque, Morphic Therapeutics, developing drugs, biologics, diagnostics, antibody drug conjugates, and antibody-directed nanotherapies. So a pretty broad experience that we can learn from in the lecture or in the presentation today. And he's worked across several therapeutic areas, including cardiology, neurology, metabolic disease, neuromuscular disease, immunology, rare diseases, infectious diseases, oncology, and fibrosis. So a lot of therapeutic areas as well. So Dr. Olivier's publications include evaluations of toxicity related to novel therapeutics in immunology and oncology, including interferons, bispecific antibodies, targeted liposomal nanotherapies, and cytokine trap technology, which I'm not sure if I know that one very well. And Dr. Olivier also, in his past, obtained a bachelor's from the University of Louisiana at Monroe and a PhD in veterinary biomedical sciences from Oklahoma State. So I'll turn that over to Dr. Olivier, and thank you so much for agreeing to impart your knowledge upon us today and be open to some questions from the audience.
Sure thing. Thanks for the kind introduction. I need to shorten that. But yeah, thanks for your time. Thanks for the opportunity through Spark. Certainly looking forward to talking to you tonight. I am largely going to focus on toxicology, but toxicology does span a lot of different areas in drug development. I often start with the three main pillars of drug development are the CMC, the chemistry, manufacturing, and controls. That's where your product, that's what your product's going to be, your drug substance, your drug product, and ultimately what you get on the market to help patients. Then you have a non-clinical pillar and the clinical pillar. And we'll talk more about how all those integrate and how toxicology supports all of that going forward. I'll share my screen. I hope you don't mind. I did recycle a lot of these electrons from previous presentations. So I try to make it fun, because if we're going to sit here for 2 hours and try to learn something, if it's fun, at least we'll have fun together and you'll remember having fun, even if you don't remember the topic or the content. It's okay. So I hope it doesn't surprise you. I made it a little scary on purpose, but the objective is, yes, keep you interested, keep you entertained. Hopefully you find something that applies to your programs and you decide you want to give it a 10. In toxicology, you've probably seen Hollywood glamorized toxicology in movies like The Rock or Aaron Brockovich, or even Civil Action to a certain extent. But in reality, it's nothing like what you see in the movies. Yes, there's a lot of things that can be really dangerous and really toxic, but it's a very serious business, especially in therapeutics, right? The number one thing in every clinical trial, even before you get into clinical trials, is safety. especially at the FDA, very conservative organization. And we'll talk about that later. I'll give you an introduction to toxicology, some boring definitions about what toxicology is, how it is integrated and essential and by law required to develop therapeutics, and then ultimately how everything kind of pulls together to get into the clinic, and then how it supports the clinical development of things to get to the market. I had to throw this in there. I do have a famous last name, I guess, for those of you who remember the good old days, I'll put it that way. But Laurence Olivier starred in a movie called Marathon Man a long time ago. And in that movie, towards the end of the movie, he keeps repeating to Dustin Hoffman, is it safe? Is it safe? Is it safe? And that's kind of what inspired the title of this particular talk. So I thought I'd throw that in there. I don't think he was a toxicologist, but maybe he played one on TV at some point. I'm just not as well aware. But toxicology is all about risk and assessing risk. Because it is safety based, we go through all our animal studies, we go through in vitro, in vivo, and in silico studies to try to figure out what are the potential adverse events. So defining what the actual events are, and then what are the potential events, and describing them as best we can. Usually it's described based on a per dose level, but we also include pharmacokinetics to describe what that dose level does, the AUC, the duration of exposure, and Cmax, how high those concentrations could get in the blood. As a result of the complexity of toxicology, I usually answer every question or start the answer to every question with it depends. There's a lot of it depends that go into answering some of these questions. So someone asked me, is it safe? Well, it depends. The formal definition of toxicology is what you see here. Again, just repeating what I said earlier, it is looking for the adverse effects, basically any chemical, physical, biological agent. So it's not just specific to the pharmaceutical industry. EPA has toxicologists, right? Toxicologists exist throughout the world. And most of the time, you have to do a lot of safety assessments, even for basic consumer products. I know a lot of animal testing has been taken out of cosmetics, but there's still safety assessments, and we still have databases that we use to predict if certain combinations or certain items are going to be toxic before you can actually market those kinds of products. The keywords here are prevention and amelioration. Most people don't think of that because toxicology is the study of the adverse events, but we actually look for ways to prevent it. Can we dose low enough, still see some beneficial effects without getting to the adverse effects? And we'll talk about how that is directly applied in pharmaceutical development. And then amelioration, is there a way we can eliminate it? If we do cause adverse effects, can we take a dosing holiday? Are there other drugs we can give to help treat any of the adverse effects, especially with chemotherapy? Chemotherapy is designed to specifically kill cells and not necessarily just cancer cells. So is there a way to help ameliorate some of those adverse events in the patients? I thought I'd take advantage of the little spark flame and add some animation to demonstrate the classic dose response curve. This, I believe, was first recorded by Paracelsus back in the late 1400s, early 1500s, and still used today. Very, very essential to establishing the adverse events, when adverse events happen and when they don't happen, and it's usually related to dose. So that by definition, right? So here's the father of toxicology. I call him Paracelsus for short because his entire name is pretty long, Theophrastus Philippus Areolus Bombastus von Hohenheim. And he basically said the dose determines the poison. So everything is toxic. I think we can be safe in assuming that. And it's just the dose that determines the poison. So if you drink too much water, that can be toxic. There's a number of other things that we can put on that list. In addition to everything is toxic, toxicology is everywhere, right? What's studying toxicology, pesticide residues on your food? Should your pets be allowed to lick your children? You know, we've got water that glows in the tub. Is that safe to bathe your? Well, maybe, I guess maybe it could be. It certainly looks like fun, so I hope it's safe. Systems toxicology, where we're actually looking at how different systems and computational biology feeds into that, where you're predicting how things are going to interact from a systems point of view. It's not just one target, but how does the target impact that cell and all the other cells in there, right? Jet fuel has specific toxicology and journals that address that, which is, I think, basically kerosene. People, right? Relationships can be toxic. So I thought I'd throw that in there for fun, but you can see there's just a lot of different avenues where toxicology exists. And even in your classroom, K through 12, we find ways to educate as early as possible about the importance of toxicology and safety assessment. Here's a number of careers. I won't spend much time on some of these slides. I think I can get through this slide deck without stopping in an hour. So please do interrupt. I'll keep watching for little messages or tips, or verbally interrupt me. I have no problem stopping and asking questions as we get to topics that you're more interested in learning about. So here's a question to be a little more interactive. What do all these famous people have in common? I'll wait a few minutes and see if someone wants to be bold and answer it. I think there's one thing you can agree, they're all dead.
They all
But
died of
they
drug
all,
overdoses. Right?
yeah, they all have a toxicology report. Yes, they died of a drug overdose. Yeah, so that was a fun little fact. But you can see that even with drug overdoses, that is certainly an unwanted adverse effect. Granted, this table needs to be updated. I couldn't find anything more recent than what I had put before, but you can see pharmaceuticals make up about 50% of the problems with US drug overdoses. So it's no small feat to make sure that these pharmaceuticals are as safe as possible. Of course, granted, most of these overdoses were because of misuse and not because they're following the approved FDA label, where tons of toxicology and clinical studies have informed the safety. They take too much of it. So again, it also goes back to the dose determines the poison. And we try to characterize as best we can what that best safe and efficacious dose is in the drug label. Of course, in the real world, we don't always have black and white, right? Not everything is going to have serious death, non-reversible, life-threatening effects, or on the other end of the spectrum, be as pretty as a rose and not have a smell, not have any adverse effects. We're usually working in this gray space of trying to figure out what organs and systems and tissues that we're impacting with our drug. And is it related to the mechanism? Is it related to the target? Is it related to physiology? There's a lot of questions that we'll ask, and we'll talk about most of those questions in this presentation to try to fill in that gray space so we can make it a little more informed if we don't have a definitive black or white situation. Importantly, this is toxicology. We do live animal studies, so I want to make sure we're sensitive to that. We've always been sensitive to that. Anyone that's ever worked with animals should know what the 3 R's are. So reducing, refining, and replacing animals. Granted, the government still require animal studies, which is why we still do animal studies, but you can see some governments such as Australia and the UK are starting to limit non-rodent use in animal studies. So we're limited to just rodents. I guess the good thing is, yes, we're sparing animals' lives. The bad thing is those animals really predict toxicology. And I've got a few slides to talk about the importance there. So throughout this presentation, we'll talk about the what, why, when, and how we do stuff. It's just a little bit about the when, so it shrinks away. So what is it? Toxicology can also in some companies be called non-clinical safety assessment. Of all the companies I've been to, I tried to pull some of that experience into this presentation because there's different ways and different organization within a company as to how you can do this, but it all funnels into toxicology. It is interdisciplinary. Oftentimes toxicology is considered a borrowing science. It's heavy on chemistry, it's heavy on biology, and it's integrating both of those expertise into trying to figure out how the chemistry is impacting the biology. But we also extrapolate. There's some risk assessment there because there's some error, false positives, false negatives. So statistics is also heavily integrated into toxicology, just like our clinical trials. We want to make sure that we power our studies good enough for the endpoints that we're measuring. A lot of our endpoints are very historical and traditional in nature. So we have tons of data on specific species that are used in toxicology on a regular basis. And that allows us to look for outliers and question the data. One of the important aspects that FDA reviewers look for is the onesies and twosies. If one animal dies, can we attribute that to the test article? If not, do we still have to worry about that when we get to patients? And ultimately, these tox studies inform the safety margin. So that dose determines the poison. I'll show you some more elaborate curves later, where we're looking for the difference between where we see efficacy, which are the benefits, predicted benefits for patients, and then where we see the toxicity and potential adverse effects. We're looking at specific organs and tissues and cell types. This is a small list of them, but typically we start with the vital organs. Safety pharmacology, which we'll talk a little bit about later. It's kind of a misnomer because it is essentially a tox study looking at the effects on your brain, your heart, and your lungs, three vital systems that if you can impact early and you can define what those effects are, if you have any effects, you might decide to go forward with a different candidate, or if the risk is worthwhile. It depends on the indication, right? So there's that, it depends. But ultimately, when we do tox studies, what's the worst that can happen? Are we really going to hurt somebody? If so, do we have to hurt them? Does the benefit outweigh the risk when it comes to treating that particular disease? Do they have any other options for treatment? How good does this work? If we do hurt them, is there a way for us to have them recover from test article related effects, but still survive the disease? Within the context of a tox study, there's a number of those historical based parameters that are very species specific. You typically have a pre-dose phase where you're acclimating the animals. You might take a blood draw here or there just to make sure you have some pre-dose baseline data for especially some of the clinical pathology, that's your serum chemistry and your hematology. So looking for white blood cell counts, red blood cell counts, your levels of ions such as sodium and calcium that are really important, or even liver enzymes. So liver toxicity is one of the #1 toxicities that you see with most pharmaceuticals. So if you can characterize that within this context of this tox study, that would be great. But you have to have some of that baseline data from the individual animals to be able to make the most appropriate conclusions towards the end of the study. On top of that, I mentioned dose a lot. The dose determines the poison. Along with the dose, we include the pharmacokinetics, or in this case, toxicokinetics. The only difference between the two is the dose levels, and that's very subjective. So we are often using toxicokinetics and pharmacokinetics interchangeably. Of course, ophthalmology is a big part of it. Do we impact the vision in rodents? Do we impact the vision in non-rodents? And then we predict whether or not that's going to translate to humans. In addition to some of these more critical, so we have anatomic pathology that becomes a part of that. That's where the gross pathology comes in, where we get organ weights, and then histology will tell us if we're at the cellular level affecting certain types of cells. If we have liver tox and we look at a slice of the liver, is it impacting hepatocytes? Is it impacting bile duct cells? All the other cell types, will do an evaluation of that within the context of the liver. Same thing during the course of the study. We have clinical observations such as body weight. We'll be looking to see if there's behavioral changes in rodents or non-rodents. There's certain tests we can incorporate, functional observational batteries, which are typically part of the pharmacology studies. We can also incorporate some of that here, but there's usually cage side observations just to see if an animal looks sick, is hunched over, isn't moving around, the activity is decreased. We can measure all of that subjectively, but trained using standard operating procedures, or SOPs, on a regular basis. For a typical IND enabling package, it's usually a 28-day tox study that's required, but it does depend on your clinical trial. What do you want to do for your first in human studies, your phase 1 studies? Oftentimes a 28-day IND enabling, repeat those study with 28 days of recovery is the most general approach, whether it's a small molecule or a protein therapeutic. Cell and gene therapies get a little more complex, and I'm happy to take questions as to what those differences may be, but we'll talk about that later. The most common species for small molecules are rats in toxicology and dogs. Most common species for protein therapeutics are typically mice, if that protein therapeutic, such as a monoclonal antibody, cross-reacts with the target in rodents. Sometimes it doesn't, and we're limited to just non-human primates as the only non-rodent species with no rodent species included in the IND enabling package. And you can see there's distinct differences in the numbers of animals. These are the pretty standard law-abiding study designs that we have. So 10 per sex per group is typically the rodents, and then 3 to 4 per sex. It's usually 3 per sex per group. And in some cases, when we have recovery, we'll add 2 per sex per group to those groups. And in most cases, the recovery only has to be in the high dose. But I usually add animals to all of the doses just in case that highest dose doesn't recover. We have a lower dose that we can test. But it depends on the study design, what you want to do in the clinic, and what the actual adverse events are that are typically described in some of the non-GLP studies before you get to this GLP IND enabling 28-day study. Most of the time, you have one control group, which is the formulation buffer, as well as 3 dose levels, 3 dose groups. And those can vary. Usually your first study, such as an MTD single dose tox study that'll be non-GLP, you'll do a half-log dosing scale. So you may choose 10, 30, and 100 mg per kg. But oftentimes you want to know from the pharmacology studies where you've looked for the benefits of your drug in some disease model, that'll give you a range of what you're targeting in the clinic, which can most of the time be between 1 and 5 mg per kg, but it really depends on the molecule and the target and the disease. I see there's a couple of chat questions. I'll pull those up real quick to address them. Discoverable data. The researcher does. So if animals die and you administered the test article, which is the same molecule that will go into humans, then yes. I don't think FDA works off of Section 8 anymore. That's the paper-based IND. They've shifted to the electronic common technical document format. So it will have a non-clinical section, which is, I know what you're getting at. That's exactly what Section 8 is. It's just called the non-clinical section, and it's in Module 2 as a summary and Module 4 for your study reports. But yes, if you have a molecule and it's the same exact molecule that killed animals that you want to take in humans, you will have to report it, and you will have to hope that the dose that I guess wound up terminating your animals is high enough that you won't ever reach that in humans, but you will have that risk following that molecule forever. And if there's no expected MTD, that's okay. I get concerned, and I have always taken the position, if you don't see tox, the molecule probably doesn't work. Even if you have great pharmacology models, oftentimes if there's no toxicity, even if it's a normal healthy animal, I get concerned that the molecule's not going to work. And I've been right 4 out of 5 times so far when I said something like that.
I have a question for you on the maximal tolerate. It kind of stems from that. And if you're looking at 2, a rodent and a non-rodent, like, how would you be sure that you're finding the most sensitive species in terms of ultimately figuring out maximum tolerated dose and then figuring out first in human dose?
Yeah, it's a great question. I think we're going to talk about that in a second, but
oh, okay.
No, that's okay. You look for a pharmacologically relevant model. You may not always find the most sensitive species, but dogs sometimes are usually the most sensitive species because you can look at a bunny and they'll vomit. So they typically, that's why they're more commonly used for small molecules, because they can exhibit adverse events at a much lower dose level than you'd expect for humans. But you have to include the metabolism, especially if it's a small molecule. There may be metabolites that contribute to the toxicity, and you'd hope that the metabolite profile in dogs matches that of humans, or at least your target patient population, so that you don't have to explore the toxicity of those metabolites. Rodents can be pretty robust, I think, just by nature and by evolution. So we have to use caution, but there are times where the rodents might become the more sensitive species. I know for dogs, and I've worked with liposomes a bit, dogs seem to be really sensitive to receiving liposomes intravenously, such that even within a minute or 2 after receiving a dose, they can become really sick or lethargic. Whereas rodents don't have the same response. They don't seem to have that response to lipid components as robustly as dogs. But that's a great question, because it's exactly why we do two species, and we try to make sure one is a rodent, one is a non-rodent, in the case that one is more sensitive than the other. And they're both mammals. All the species we use are mammals, so they're very well conserved endpoints from a tox point of view that will translate to humans. It's just about finding where that dose level is and where that exposure is that results in adverse events and doesn't.
And I have one other question. And in terms of unanticipated adverse events, like from the get-go, in terms of like immune mediated reactions, I mean, it's hard to predict that. And there's no requirements right now for any kind of like a humanized rodent model or something. So how do you handle that currently? If you expect that your compound, based on its mechanism of action, may have some implications to the immune system, for example.
Yeah, that's a great question and a big concern. And I think we'll continue to be challenged with finding a model that can directly address that. Right now, the assumption is immunogenicity in animals does not represent humans, just because there is so much variation in how the immune system responds to various chemicals in animals. It just doesn't predict. Even within humans, doesn't predict for humans. So we do use more in vitro data. And as you mentioned, the mechanism of action studies, as much as we can explore a mechanism of action of our molecule and its effect on the target at the cellular level, or even at the tissue level, or if you have some kind of model level, that's great, because there's oftentimes tox studies can't predict it. The challenge with humanizing mice is the immune system is human, but everything else is not human. So it's hard to directly compare exactly what the immune system is going to do to everything else in a human if you just use a humanized mouse.
I think there's another question there too.
Yeah, can you see when I pull up the chat window? Otherwise, I can read these questions off. Right now, I have the chat window open. I don't know if you can see it, if you just see my slides. I'll just read it. Richard Dukas to everyone. As an example to support Dr. I guess Glode's question, I hope I said that right. Suppose you're developing a cancer drug that targets a pathway that isn't present in healthy cells. Say tumor cells are less sensitive in nanomolar range, but normal cells aren't sensitive at any pharmacologic dose. I think that was just a statement though. So hopefully that was a good statement. Let's talk a little bit about the whys of toxicology.
Can I
yeah.
just follow on that quick, Ken?
Oh yeah, sure.
So I
Yes,
think
sir.
he was asking about no expected MTD. So in a situation where either a fetal antigen or something else is the target for your drug that's targeting this cancer cell, there may be no real normal cells in the body that are actually sensitive to the drug. So how far do you, knowing that if you push the MTD too high, the FDA at some point may say, well, you didn't have an effective, so you have to go as high as you can possibly go. I think that's what he was, I think that was what he was asking with his question. And I know we've got a lot of oncology projects that occasionally are either locally delivered products or products where normal cells do not really, the drug may or may not have much of an effect on normal cells or healthy cells.
Okay. Yeah, thanks for the clarification. Yes, if there is an example where a target is almost exclusively expressed on a cancer cell, or maybe it's just at such low levels in normal cells, then maybe it's not pharmacologically relevant, but you would be pressed to do a maximum feasible dose as opposed to a maximum tolerated dose. That just means, to Richard's point, you push the dose, or you increase the concentration so much in that injection site that's as high as you can go, and that'll still give you coverage for what you want to do in the clinic, like when you go subcutaneous or intracranial. Well, there's a number of other routes, but let's just stick with subcutaneous. There's only so much volume you can put in one injection site, and so you would choose to go at the highest concentration your formulation will allow for clinical application, and inject subcutaneously as much of that as possible. In animals, we can make some exceptions and maybe have up to 4 different subcutaneous injection sites, but in rodents that becomes limiting because you can't have all these subcutaneous injections on the animals. It's going to be adverse in and of itself. So we just use caution and use judgment when we're doing those studies and pushing the maximum feasible dose. And then that's where you have the conversation with the FDA. Is like, we didn't see any toxicity, but we understand the expression profile in normal cells versus the expression profile in our target tumor cells. Here's the maximum feasible dose. We still feel like we can come in 5-10-fold lower than that to be able to demonstrate the benefits to the patients. But we won't know for sure all of the adverse effects until we do our phase 1 dose escalation trials.
So Ken, this is Bill Brack, and I have a background in toxicology as well. I guess I just, in addition to sort of the caveats you've mentioned, is comment that in general, if you look at ICH guidance, FDA guidance, there is kind of a maximum dose, assuming you can actually dose high enough, and there's no maximum feasible dose limiting you. A dose of like 1,000 to 1,500 milligrams per kilogram is kind of what they define as maximum. And if you find that and the other part of that is, I think you minimally need to be able to achieve a 10-fold margin over what you're expected to dose in the clinic. But again, there's some caveats around that as well. So.
Yeah, good points, Bill. Thanks for sharing. I just thought this picture was humorous. I hope I never experience a flight like this, but I'm sure people have, and looks like in this case they did. This looks pretty real. Nevertheless, the job of the toxicologist is to identify those hazards and then risk assess those hazards. So if you can't identify maximum tolerated dose, I guess it's less consequential from toxicology, more to demonstrate that you do have some active component going in there and that it should have an effect in the patients. But more so if something looks toxic. So some of the in silico evaluations that we do using software such as the Derek Nexus software, that is basically an archive of tons of chemistry that started out to predict carcinogenicity. If there were similar chemistries that you could look at, like benzene, for example, you could predict if anything had benzene in it, it was most likely going to be a carcinogen. Similar things like that we try to do in silico or even in vitro to predict what those hazards might be, and then try to test them out as best as we can with animal models before we get into humans. I thought this sign was kind of funny. Do not visit if you are ill. This was on a hospital. Help protect our page. So I think I took it out of context, but nevertheless, it was just funny to see this kind of sign on the hospital. You could see everyone's very safety conscious and very protective of not just hospital employees and patients, but even workers. So whether you're a DC fan or a Marvel fan, when it comes to comics, it's all about protecting our patients, making sure that we're defining the benefits and the benefits outweigh the risks going into the patients. Another funny cartoon, FDA says, if laughter is the best medicine, we're going to be regulating it on Monday. That sounds very much like something FDA might actually say these days. But in addition to the FDA, there's OECD, there's the Ministry of Health, Labor and Welfare, European Medicine Association that also regulate drugs to a very similar standard worldwide. So you can get your drug approved in one country and those agencies talk to each other. We go to the same meetings. I have friends. When we graduated, they went to the FDA. I went into industry, and most FDA reviewers may even bounce around in consulting a little bit after they spend some time at the FDA just to try to help out more companies as much as possible, which is great because then we can all try to communicate with each other and set the best standards for our patients and what kind of safety we want going forward. And Bill mentioned ICH, the International Conference on Harmonization. That's kind of how these regulators get together. I'll mention this a little bit later. So as a toxicologist or just as a human being, I'd love to have a crystal ball. I have a feeling this is what my crystal ball would look like, very cloudy. I wouldn't be able to predict anything like Harry and Ron here. Also would like to be able to talk to the dead and learn lessons learned, right? Learn from our forefathers as to what worked and what didn't work when it comes to toxicology and life in general. But that's just not the case. So we have to make some general assumptions, much like we do with statistics. We have to assume certain things will happen or not happen, will go a certain way. And animal models to date, I'm convinced that they're still the best way to predict the potential toxicities in humans, and then going to as high a dose as possible. That 1,000 or 1,500 mg per kg that Bill mentioned is really critical, that we can explore when does it actually get toxic, and if it never does, why doesn't it get toxic? And so there's publications that explore this in much more detail. Probably the key publication is explained here, where they're looking at the ability of animals to predict the toxicity in humans. And so Olson et al. Published a paper saying that 71% of the time we can predict the target organ toxicity in animals that's seen in humans. And there's a 94% of those toxicities can be detected in that 28-day study. Here they have it as a 1-month study. So that's why that 28-day study becomes pivotal for the IND, because you can predict 94% of the toxicity 71% of the time. There's no other way. We can't even predict the benefits in humans that good, which is excellent because we want to know what the adverse events are. We really want to be able to keep our patients safe as possible. So having this level of standard, it's going to be really hard for anything else, including artificial intelligence, to beat actual biology, the biological results from these toxicologies. And this is published from a reputable source, you'll see. To get more into the detail, there are challenges when we're trying to translate the data from animals to humans. You can see I've got set up here what the subject information, the doses, circumstances, and procedures that we're using from animals to humans. In our toxicology studies, we must use normal, healthy animals that have been well characterized, they're well behaved, they're well maintained, right? They're species that have been purpose bred for hundreds of years in most cases. Whereas in the human situation, it's a patient. You know, we're trying to use data from healthy animals to predict for sick people. The challenge is, well, in a healthy animal, we can have very tight specifications for our toxicology endpoints. If we introduce a disease in the healthy animals, well, that disease itself can perturb some of those endpoints. So we need to eliminate every other variable except for the test article. The test article and various dose levels of the test article compared to a concurrent control is the best way to figure out if an adverse event is test article related or not. Is it dose dependent or not? Is it recoverable? Is it reversible, and where it happens? Whereas in a sick human, that's where we're taking our chances. We still generate the best data and the best safety margin possible with healthy animals. And as also Bill mentioned, sometimes we have that additional safety factor of tenfold. It's not always the case. It depends on the indication. It depends on the urgent need of the patient. So the FDA and scientists in general are very flexible because we want to make sure we accommodate, right? It's got to be fit for purpose and suitable for its intended use, which is the whole point of having labels on our drugs. So we want to make sure we have the appropriate safety margin. I've typically had 3-fold or less of a safety margin going into cancer patients, but it just depends on the mechanism of action of the drug, especially chemo. Most of the chemos have no safety margin because they're specifically designed to kill every cell it comes in contact with. You just hope that it kills all the cancer cells before it kills the patient. Another thing with nutrition, right? Purina makes rat chow, they make dog chow, they make every kind of chow. So it's very regimented. It's percentages of proteins, fats, and carbohydrates on a regular basis. Oftentimes it's in solid form, so they really have to work for it, especially for rodents, to make sure their teeth don't keep growing through their lips. And then with humans, it's variable. Sure, we can control it as much as we can in the hospital, but a lot of sick people are taking pills and they're not always in the hospital, so we can't control what they eat. And I know if I was sick, sometimes there's certain foods that make me feel good, but those foods are not good for me. So there's a variability there. And if it's an oral drug, this will usually dramatically impact the availability of your drug to get into their system. That's why oftentimes drugs will have do not eat before taking. Some cases it says to eat before taking, and that could be because it's a tox endpoint that said it's too irritating to the stomach lining. You have to have some food in there for it to work well. And then most importantly, the histopathology. This is probably one of the most critical endpoints in toxicology because we're looking at tissues that have been harvested from these animals that have donated their lives and their tissues to science so that we can explore and slice them up and look under a microscope to see at the cellular level what cells are being impacted and what kinds of adverse events we can predict based on the cell types, based on the organs and tissues that we look at. Can't do that in humans, at least not ethically. And just another brief list of some common human reactions to drugs. I guess one superpower I'd like to have is to be able to talk to animals, maybe call it Dr. Dolittle syndrome, because you can't ask an animal if they're depressed. You can look at their behavior and try to predict if they're depressed, but you don't actually know if it's depression. Other things like hypotension and constipation, a lot of other things, weakness, are very challenging to be able to translate from animal studies to humans. So when it comes to humans, right, pain is something that's tough to measure. In animals, you measure reflexes to pain, but you can't ask an animal if it's in pain. Nociception is, I guess, a somewhat controversial topic to some as well, as to whether animals actually feel pain, depending on how many receptors, what receptors are present. But trying to figure out if an animal can hear ringing in their ears, heartburn, all these other things. These are some of the most common side effects that you see in clinical trials that just cannot be measured in animal models yet. So let's talk a little bit about when do you do toxicology? You'll see from some of the graphs I have, toxicology is prevalent throughout the whole drug development paradigm, as well as even post-market. While you're on the market, that's when you're getting into the broader patient population. Doctors and patients are having conversations, maybe using your drug off-label. We want to make sure we can accommodate as many different adverse events as possible, even if it's used not for its intended purpose, which is the label part. So we try to follow the ICH that was mentioned earlier, the regulatory guidelines that cover all the world's drug development. But strategic development, when are programs coming into the pipeline? When do you want to make a decision on your lead candidate? Sometimes you want to do some early screening toxicology just to confirm which of those development candidates will become the clinical candidate. Some might have more adverse events in the cardiovascular system. Some might have potential to target the nervous system. We just want to make it clear which is the lead and which is the backup, and have as much information about the potential safety concerns as possible. Of course, when you do it is contingent on when you have a clinical plan. Once you find an animal model of pharmacology that represents as much as possible the disease state in patients, you can formulate your clinical plan, find some dose levels and exposures that seem to work well, and then move into some early tox screening. And of course, every time you do a study, there's a reason why you did the study, to test your hypothesis. And then what's your conclusion based on the data that you gathered? Hopefully you pick the right endpoints to be able to generate enough statistics to either reject or fail to reject your null hypothesis, which means there's no effect. And then that will inform the future studies. And now the how do we do that? So here's the more elaborate figure that talks about a therapeutic range versus the toxic range. Oftentimes in our tox studies, we're claiming to try to define the no observed adverse effect level, the no AEL. Oftentimes you may also hear of a no EL, no observed effect level. There's a number of other acronyms out there, but the most common one that we use to set our starting, or at least estimate our safe starting dose in humans, is the no observed adverse effect level. So we can still find that there's some effects of your test article, but they're not considered adverse. So there's a lot of subjectivity looking at that histopathology, looking at your clinical pathology and all the other aspects that we talked about on the previous slide. In totality, what do all the data say? Is there a dose level where there is no observed adverse effect level? We set that and we'll define that in the course of the study. We'll associate it with our pharmacokinetics, usually our AUC, area under the curve, as well as the Cmax, and then your margin of safety. You hope that you get a graph that looks a lot like this, where your therapeutic dose range from your pharmacology studies is here, and you go 100 times higher before you see toxicity, but oftentimes it's very small, if not absent from your drug development paradigm. So in conclusion, the risk assessment that goes along with toxicology, there's no real fixed rules or recipe books. There's a lot of guidance out there. from specific agencies in the country you're interested in, as well as the international conference that gives you the general guidance on safety. They don't tell you exactly how to do the studies. They just tell you some of the expectations. They may throw out some sample study designs, but you want the guidance to be flexible. You don't want the guidance to be too rigid because there's so much variability. There's so many indications out there. It's tough to have a guidance to specifically tell you exactly what to do. And I guess that's to the toxicologist's advantage, because we can come in as experts and just use all of our training, education, and experience to help guide how that program looks from a safety perspective and integrate it into the overall development plan. Ultimately, the key endpoints should address reversibility. So as I talked about, if there's a test article related effect, you want to know if that effect goes away once you stop dosing. So that's why that 28-day IND enabling GLP study often has a recovery phase, a drug-free, a dosing-free recovery phase. So you can see if the animals that you took down at day 28 match, or if the animals on day 59 get better. Monitorability, that's technically not a word, but the FDA uses it, so we can use it, right? The government makes up words we can use. Monitorability means that you can easily monitor it in a patient. Hopefully it's non-invasive, but most of the time we try to correlate the clinical pathology, so your hematology and your serum chemistry, with the histopathology. So if you've got liver damage, you should see elevated liver enzymes. You can use that to predict whether or not it's test article related and if it's monitorable. And in patients, that'll translate well. So if we can get the time frame down in rats, if it happens 1 hour after dosing, we see an increase in liver enzymes. By 28, the liver looks like it's somewhat necrotic or apoptotic. We can use that to say it's a potential adverse effect in humans, put that information out there, and then the doctor and the physician, the patient can have a conversation about whether they feel like they want to take that risk on, if the benefits outweigh the risks for them at a very personal, individual level. And then we've already talked about the safety margin piece. The interdisciplinary is very important. It can't just be one person looking at a study and deciding what the NOAL is. It has to be a pathologist, a toxicologist, a veterinarian, a study director. There's usually 3 or 4 people involved in these decisions because a lot of the decisions are very subjective. The data, the histopathology data, is looking under a microscope in a field of view and deciding if 10% of or less of a particular field of view has that finding. If it's a foamy macrophage, for example, you have to look and see at several different tissues, several different animals, and decide if that is actually an adverse effect or not. Because it's not mechanistic, right? It's just morphology. We talked about this a little already. The pharmacologically relevant species is really key to toxicology. If the species of interest doesn't express the target, or it doesn't have any relevant biology that's going to translate to the patient situation based on how the target and the molecule interact to result in the physiology, then it's not considered physiologically relevant. We won't be able to use that model to predict adverse events. I've already talked about this, but just quickly, rats and dogs are most commonly used for small molecules, non-human primates for protein therapeutics. To a certain extent, gene therapy has been using non-human primates. But cell therapy has an even bigger challenge because we can't use normal healthy animals. The way the T cells or the CAR Ts or TCRs or whatever cell therapies you're looking at work is you need the intact disease as well as the intact human immune system. The humanized mice can work to a certain extent, but as I mentioned before, they don't have all human tissues. So you don't know if you're going to hit off tumor or off target items unless you have that present. So we've restricted a lot of the cell therapy toxicology to hybrid models. We call it a pharmacology toxicology study. It's basically a pharmacology study, so it is a disease model. And then we tag on some of the toxicology endpoints that we described earlier. And we just hope that the disease doesn't perturb those tox endpoints to the extent that we can't definitively say if it's a tumor related or if it's a test article related. That's the biggest challenge and limitation with some of the animal models in the cell therapy space today. And the FDA understands that. We filed 2 INDs on the same day, December 20th last year, for TScan. And the FDA comments corroborated the fact that, yes, we understand the limitations of live animal models. And then, as I mentioned before, you rely more heavily on in vitro mechanism of action data. How does the T cell interact with tumor cells? And then how does the T cell interact with normal, healthy human tissue cells? And we'll do both of those in screen. And it does contribute to decreasing the number of animals that we're using. So that's one stride forward. When we're designing the tox studies, we want to make sure it matches the clinical environment as much as possible, especially the route of exposure. We can't develop an intravenous human drug without doing intravenous animal studies. Same with oral. So you want to match whether you're going oral, intravenous, and these are various formulations. Some drugs can't be given oral. Most protein therapeutics are given intravenously just by nature if it's a monoclonal antibody. Otherwise, it gets digested in the GI tract and it doesn't get absorbed. So most of that's given intravenously, but certainly small molecules still make up about 70% of the drugs being developed out there and can be very effective if given orally. And nowadays, there's such advanced artificial intelligence and systems where you can 3D model the actual structure of not just the chemistry, but also the pocket, the binding pockets, the protein pockets, the crystallized structures where those molecules can interact. And I've even walked into dark rooms where the chemists and the biologists had their VR headsets and they're moving molecules around to see which one fits best in the binding pocket to determine what the chemistry should look like. It's pretty fascinating how things are coming along. But that's a good way for us to move forward with small molecules and make them more soluble, make them more permeable, and then make them get to that pocket in more individualized, specific type medicines. I thought this graphic was kind of neat in the animation. But nevertheless, when you're designing the tox study, you pick, as I mentioned, 3 different dose levels. The low dose you want to pick, usually it matches the clinical dose level or it matches a dose level in the pharmacology studies that are usually an 80-50 or an optimal efficacious dose. The mid dose, you want to start to see some signs of toxicity if possible. And then, of course, the high dose, we're toxicologists, right? We're not trying to hurt animals, but we're trying to figure out what's the worst that can happen. So we really want to push the dose into that, 1,000, 1,500 mg per kg. Sometimes I've gone up to 2,000 just for a single dose. And then you just start reducing the dose levels as you get into the repeat dose phase. Just checking for questions. Great. It is reams of data. When you get one tox report, it could be over 2,000 pages, but of that 2,000 pages, 90% of it is raw data. So you can imagine if you've got, 10 animals per sex per group in rats, and you've got 4 groups of that, you're talking about hundreds of animals. And of those hundreds of animals, you've got time points for pharmacokinetics, you've got time points for clinical chemistry, you've got a couple of time points for anatomic pathology, but you've got 37 to 39 tissues from 400 different animals. So you've got a huge amount of information captured, and it all has to be written up and summarized and test article related or not, monitorable or not, reversible or not. All of that information comes out in the written form just by looking at all of the data on top of all the statistics. Then you have to talk about if it's statistically significant, is it also biologically relevant? Those kinds of conclusions come into play. Differences in species, we talked about differences in sensitivity. That is key. Why is 3 mg per kg toxic in dogs, whereas 30 mg per kg is toxic in rats? Why is there a difference there? Sometimes it's metabolism, sometimes it's target expression, sometimes it's just robustness of the rodent model not being as sensitive as a dog to some of these endpoints. And then, is it safe enough? That's where the safety margin comes in. And then, of course, I say educating regulatory agencies. Hopefully there's no regulatory agencies on the line. I don't think they'd like it put that way, but we all have to work together. I have to learn from the FDA what their expectations are, what they've seen that they can't share confidentially. A lot of at the IND stage, none of that information can be shared. It has to be confidential so the FDA can hint or can recommend things without having to justify it, which can be frustrating to a scientist, right? Because I want to know why you want me to do this, and I can't tell you that. And then same thing on the other side is I'm a scientist. I'm coming in with this novel therapy that you may have never seen anything like before. And I have to educate you on what that molecule is, what the intended therapy is, what the indication is, and then why we think it's safe, and all the studies and the data that we're using to justify the safety.
I have a quick question. If you have an IV, like let's say you started with an IV formulation and you did all your testing in animals and IV, and then say that that goes onto the market and you have all that data, and then later, you know, I don't know, for some reason you want to go with an oral formulation, like how much do you need to provide in terms of like GI, for example, since it's going through, you know, through an absorption, and that for in terms of safety, like how much do they require if you're changing the dosing formulation to an oral dose, say, or some other formulation that's, you know, other than IV or like sub-Q, like something other than that?
Yeah, that's a great question. The good thing about intravenous is there's no absorption phase, so you just assume it's 100% bioavailable. It's getting right into the bloodstream and getting distributed, metabolized, and eliminated. So you don't have to worry about some of the other barriers. It also maximizes what your maximal concentration in the serum or plasma is, depending on the entity. So you'll know at that dose level how high the concentration is in the system and how it's getting distributed. The other part to intravenous is you get the duration, right? Sometimes the metabolism can be different going oral. You go through first pass metabolism, so you might lose half of your drug just because it has to get absorbed through the GI tract, and the liver is going to filter out half of it or metabolize it and eliminate it before it even gets into the system. So you maximize your exposure with an intravenous route. You can still use that data to support other routes. The difference is you should match the route because there could be something related to oral administration that has an adverse event. Maybe there's something irritating in the stomach that happens. You can't evaluate that with an intravenous route. So you could do what oftentimes is called a bridging study. It just depends on what your clinical plan is in patients. You want to match that as exact as possible. If you started with intravenous and switched to perioral, you might have to do some additional, as you mentioned, formulation work. And if the formulation is more aggressive, you might want to look at the excipients that you're using. Now, hopefully they're non-novel excipients, and there's a list of what's novel, at least what's not novel to choose from. And there's usually some strategies about how to formulate things with less aggressive solubilizers, for example, so that you don't have that irritation effect. Because then you have to try to tease out what's the formulation related effect versus the test article, which is why we control for everything in those tox studies. So a bridging study might be a minimal thing that you could do to explore the pharmacokinetic differences, which would be the exposure differences, as well as the potential local tolerance type effects if you go oral versus intravenous.
So are there specific guidance that would be required for the tox perspective though? I mean, I understand the PK piece, but the tox perspective, if you were going with that to kind of do any kind of tox that was related to any kind of like GI, for example.
I don't know if there's specific guidance that talk to routes, but I know in the M2 guidance for ICH, it's a multidisciplinary guidance. They just recommend if you're going to change routes in the clinic, you should also do the comparable tox studies to support changing that route. It doesn't mean you have to redo your entire tox package. That's why the bridging study is a compromise. But maybe you do. Maybe there's no way to do a bridging study unless you repeat everything using the oral route versus the intravenous route.
So Ken, this is Bill. I can help you out here because I've had
Yeah, sure.
specific experience in this area. There
Great,
is an
go for it.
there is an FDA guidance about non-clinical safety evaluation of reformulated drug products. So, and as you indicated, you know, you're really focusing on the specific route that you're changing to and trying to assess those toxicities. But yeah, the FDA issued a guidance. It's the only one I'm aware of. ICH doesn't have anything specifically in this regard, but it's accessible.
Reformulating, okay. Yeah, that's good to know.
And if you want it, I can share it with you or share a link either way.
Yeah, share it with the group. I have done some reformulated stuff, but it usually is done before I get into any talks. But that's good to know. Yeah,
Yeah,
thanks. See, we can all learn from each other. Yeah, I appreciate that.
Yeah. Well, the interesting thing is now with a lot of drugs being looked for other types of indications than what they were originally approved for, especially drugs from long ago, you're seeing different routes and different opportunities developed for those. And you don't really want to have to repeat the full package.
Agreed. Here's just some quick reasons why you may not have seen that effect in non-clinical studies, right? Animals don't always equal humans. That's the assumption slide that we talked about earlier. Statistics is a bit of a number game. The more numbers you have, the higher the power of the study to predict whatever the endpoint is. That we have a lot of control over the dosing in the animals. We don't necessarily have that in humans. You see all those people overdosing, right? We can't control what a human's going to do. It's a very personal thing that the person has to decide, have a conversation with their physician, and hopefully they're going to make the best choice for themselves and everyone that loves them. In vitro doesn't always equal in vivo. In vivo results usually trump in vitro, but if you don't have good in vivo results, like with cell therapies, you have to rely more heavily on the in vitro to predict potential toxicities. Males and females are just keeping it to the two sexes in the context of genetics here. Exposure can be different, right? There's sometimes differences, different levels of enzymes that might impact the metabolism of your drug. So you'll have to be aware of that. We always have males and females on every study. And then of course, rats don't always equal dogs, don't always equal primates. You have to make certain assumptions. You just assume that those well-conserved tox endpoints that we measure in mammals also translate to humans. So we'll move into the therapeutic development piece after covering toxicology. I found this amusing, that I wonder if this is how we actually viewed what a headache was. Sometimes it actually feels like that, right? There's a little trumpet in the ear. I thought that was, anyway, but this is more real, right? We're the expression of things going up and down, and neurotransmitters and transmembrane receptors. So we've gotten way more sophisticated in how we think about things. I guess enabling us to go to the moon, right? And to go into space again. And now people can just pay to go into space. So it's just amazing how evolved humans are. Anyway, when you think of drug development, I can put it in the same context as getting your paper accepted. I think there's more academicians on this call than I usually have. So I know what it's like to get papers approved, and even to today, getting things published is going through the gauntlet, right? Going through the gauntlet of scientists and publishers and editors to get what's out there. And you just hope that your paper doesn't lose the original intent and the conclusions that you had at the beginning to be able to get out into the public domain. Same thing with the drug. You have to go through all of these various stages of drug development, called the gauntlet, to get to where you can actually launch it and have a benefit. This means when you're marketing your drug and the FDA decides to approve it to patients. And you can see over the course of many years, usually 7 to 15 years for just the clinical development piece. And the discovery side, it could take another 5 to 7 years, depending on what's known about the target. Going through the various phases of development to get to the 1.5 years of FDA sifting through your 400,000 to 500,000 page new drug application or biologic license application before you can get that to patients. And this also, as you'll see, it contributes to the cost of everything, so that billions of dollars that you see, I think these days it's probably not 1.2, but it's probably closer to $2.5 billion to develop a drug. It's because you're paying for all the failure, right? 5 to 7 years in discovery and another 7 to 15 in clinical. That's like a lifetime of failure. You really have to be ready to fail often and a lot in drug development. Not to discourage anyone, because we need those innovators out there. But that's the challenge for me is, how do I make sure something's safe, that it's not going to hurt somebody, and focus on the day-to-day operations as opposed to the 15 to 20-year failure paradigm. You've probably seen something like this before, but I just spoke to this. You're going to fail a whole lot in drug development. And I just thought this was funny, right? The weather people fail a lot too, but I think we actually beat them in how often we can fail. It's like coming up with the light bulb. You got to fail 10,000 times to find that one molecule that's really going to help somebody. And hope you get it right. Reasons why we fail, thankfully, it is live animal studies. It's unfortunate that we have to use live animals. Anything alive is precious, plants and animals included. But if we're going to keep human species as a priority, then we want to explore as much of that as possible. At least we give the animals a great life while they serve mankind. And in this case, animal and safety tolerance does predict the majority of candidates that are not safe, so we can stop it there. The important point we can make here is going back to that Olson paper where you had, was it 91% of toxicities predicted 71% of the time. This contributes to that. It would probably increase that concordance if we had some of these candidates that were killed early in development move into human studies. The only thing that paper considered was candidates that went from the animal studies to the humans and predicted toxicities that were not lethal. So in this case, that's a good prediction. Oh, this is chat. Oh, that's the link. So thanks, Bill, for sending that link in the chat session. Here's just another way to look at that. What are some other reasons why candidates get terminated? Toxicology still makes up the bulk of it in clinical. So the safety makes up 40% of why things are being killed. Efficacy is typically number 2, but certainly safety. So this will be your proof of concept if you move into your phase 2 studies or a study where you're actually looking to demonstrate the benefit. It's still a large proportion of the safety that impacts why candidates stop moving forward in development. Here's some of the key players, just if you're not already familiar with how to build some of these companies. Since I've left Miramac, we built that company over close to 12 years. I was the only toxicologist on every project we had, which is where I got a lot of experience in monoclonals, bispecifics, liposomal encapsulated small molecules, nano-targeted therapies. And this is how we built the company, and it matched a lot like how we're built Regeneron and how Biogen existed at the time I joined. So the main components are the CMC, the non-clinical space, which is a lot of what's covered here in the clinical space. And in every case, there's regulations. So the CMC is the chemistry, manufacturing, controls. This is probably the most important part of everything you do because this is your product. This is what you're ultimately going to sell to benefit our patients. Comes under good manufacturing practices. All of your non-clinical studies, including assay development, pharmacokinetics, toxicology, all of this, everything with the exception of pharmacology, comes under good laboratory practices. And then finally, your clinical comes under your good clinical practices. So again, this reinforces that 3 pillars of drug development. All of that is heavily regulated, no matter what country you go to. I think US is number 1, followed closely by China. The good thing about China is they are copying a lot of the regulations, almost word for word, that the US uses, as well as the European Union and Japan, probably the main regions that actually develop drugs. This is just a brief outline of how the drug development paradigm works. So as you start off early, you're looking for some early safety predictions. You go into your standard non-GLP, GLP toxicology to enable your IND. Once you have your clinical trials ongoing, going from your first in humans to your proof of concept studies, you may have to conduct additional chronic tox studies. So 3, 6, 9, in some cases, 12 months tox. So that means you've got a year worth of tox in your non-rodent species. Most of the time, rodent species, the most chronic you would do is 6 months. But you have to get all that data depending on your intended use. What's your label? If you're going to be an acute treatment, you may not have to go this far. For example, in oncology, the most chronic tox study, the longest chronic tox study you'd have to do is 3 months. And that is also in the guidances, ICHS 9 specifically. And that's just because patients typically aren't living long enough to be on a therapy that long, or the therapy is so toxic they can't take it for more than 6 weeks at a time. So you don't need to have a ton of toxicology to support some of those. As you get into women of childbearing potential, reproductive and developmental toxicity, or if you flip that, it's DART studies, developmental reproductive tox studies, are conducted, one mainly to protect the embryo, the thalidomide tragedy of a number of years ago in Europe. Fortunately, it didn't happen in the US because FDA reviewers saw what was happening. As I mentioned, these regulators talk to each other all the time, even back then, and they'll see what new drugs are coming around in different countries, and they'll want to see, hey, could this benefit our population in our country? And so fortunately, we avoided the thalidomide tragedy, but of course, that's going to resonate now through history to make sure we do not repeat that. As a result of that, we have developmental reproductive tox studies that we perform usually in rats and rabbits. Those are the top 2 species, but there may be times, as I mentioned, where your compound does not cross-react with rodent or any other species besides non-human primates. So you do a reproductive study in non-human primates. There's a few segments to that, but we can talk about that some other time. And then, of course, once you launch your product, as I mentioned, you get into that broader population, the bell-shaped curve of adverse events takes over. And you may see things that were not predicted in clinical trials. You may see things that were not predicted by tox studies. So you just want to use your pharmacovigilance to track all of those adverse events that are being reported and find out how real they are, how test article related they are they monitorable, et cetera. And if you have to do any further tox studies while you're selling the drug. This was a matrix environment just to try to show you how teams get formed in some of these companies. Oh, there's a couple of chats before we get into that. It looks like the link in the chat isn't functioning properly. We can follow up with that after the call. But in a matrixed organization, this is just a way to look at when someone has the leadership. You can see based on this, the L shifts from team to team, which is on the y-axis here. The x-axis at the top is just different stages of development, where your early research, you're in manufacturing, movement to formulations, assay development, toxicology, ultimately getting into the clinic, filing the IND, and then commercial assessments before you put your registration. The leadership kind of changes, right? So early in discovery, we're waiting for discovery to discover a molecule that impacts the disease. Once we have that molecule, we move it into manufacturing, and their expertise takes over to manufacture it, scale it up, make gram, kilogram quantities of the molecule. And then it has to be formulated. You have to develop assays, both on a quality control from a CMC perspective, as well as bioanalytical assays to support your pharmacokinetics and anti-drug antibody if you need that in your in your toxicology studies, and in your clinical applications. You'll have to have the same pharmacokinetic type assays ready to go. So the leadership changes over the course of time, depending on the stage of development. This is just another way of looking at it, right? You have money to fund the research, to make the drug, to ultimately get it into the clinic. This was a cool picture that I found in an archive, and it shows you at an FDA reviewer's desk just how many pages some of these NDAs would have. It's just like 400 or 500,000. Now this is a paper submission. As I mentioned before, FDA has moved away from paper submissions to electronic. So it's much easier and not as burdensome on our forests to produce some of these documents. But there would literally be truckloads of paper that get loaded up at GSK and shipped over to the FDA for the reviewers, because you're looking at sometimes 10 to 12 different reviewers, because you've got CMC, non-clinical, clinical reviewers. In those cases, you've got a lead reviewer, you've got a supervisor, and sometimes you've got 3 people for each of those. So you could have as many as 12 different people that need a copy, and FDA sometimes will request 12 to 15 physical copies of your application. I haven't seen that much lately, but there's still an opportunity that could happen. least at the pre-IND stage, I think I've had that happen in the past couple of years. This is just to demonstrate what that electronic common technical document would look like. It does have a specific type of window. Now you can tell this is pretty old and this image is very blurry, but you can see the file folder structures. There is more definition of what the common technical document is. If you go to the ich.org website, you can look at the guidances that talk about the common technical document. You click on the M for multidisciplinary guidances, and you can look up well, it's in the title, the common technical document. I don't remember the exact number off the top of my head. But ICH also has the common guidances to cover safety, which is the S guidances. That's all the GLP or non-clinical guidances. There's the E guidances cover efficacy. That's everything that covers good clinical practices and clinical ethics. And then there's the Q guidances, which stands for quality. That's all your CMC guidances. So again, sticking with those three pillars, the CMC, the non-clinical, and the clinical, all have their own specific guidances at ich.org, as well as M to cover multidisciplinary aspects. There's a few more chats here. It's just figuring out that link issue. Great. Any questions while I take a breath? Hopefully I'm not talking too fast or too much. Lending itself to that common technical document, this is the structure. So this is what they call the triangle. So you've seen the food pyramid. This is just the CTD triangle. There's different modules. It's 5 distinct modules. The first module is regional. So if you're filing your IND in the US or your NDA BLA for that matter, this is designed for your registration enabling application. But if you start in the common technical document at the first in human study, then you just keep building out everything in common technical document. You don't have to go back to any other types of revisions when you get ready for registration. So if you went in the US, everything in module 1 would be US specific, meaning the United States has forms like a form 1571 is a 1-page form that you fill out just to say, hey, FDA, here's who I am as a sponsor. Here's our clinical trial. Here's what we want to file in the IND. It's just the paperwork part. 1572 is the form that your clinician as a sponsor would sign off saying, I'm accountable and responsible for all the patients that get enrolled on this protocol for this trial, for this indication with this molecule. When you move into module 2, these are the more critical pieces because this is where you have the overviews of everything that's happening in modules 3, 4, and 5. Again, those 3 main pillars of drug development. The quality overall summary is your CMC summary captured from your quality documents in module 3, which is everything you're doing in manufacturing. Tons of flow charts, tables, certificates of analysis, testing, the QC that's going to happen, all the quality that goes into your final drug product. But you also talk about your drug substance and your manufacturing process and formulation, everything that goes into making material. It focuses mainly on your clinical material, but you also talk about the non-clinical material used in the tox studies. In the non-clinical overview and summary, you're talking about your pharmacology, your pharmacokinetics, and your toxicology. Those are the 3 main pillars of non-clinical drug development, everything you would need to support an IND and registering your drug. And then finally, the clinical. In an IND, if you use this structure, there's really not much substance here. You have your clinical protocol, you have your informed consent, and you have your investigator brochure. And those kind of go in as attachments. The investigator brochure goes into module 1, but you can tack on your protocol and your patient informed consent. All 3 of those documents have to be reviewed by the regulatory agency. They also get reviewed by your institutional review board, but I guess that's a talk for another time to cover clinical. Are there any advantages or disadvantages of performing pilot PK tox studies in the university? Yes. 1 big advantage is time. 2, cost savings. It doesn't have to be a GLP study to evaluate safety. You can design a tox study anywhere, and it will always contribute to safety. You just want to use caution when you're doing a tox study anywhere, even in a university, because if your molecule is eventually something you want to take into humans, carefully design your study such that you can conclude whether or not it's test article related or not. If you come up with a study and you give even the lowest dose level that harms the animals or kills all your animals, you're kind of stuck with that data forever. And I say stuck with it in a bad way, but it just means to use caution when you design your studies, that you're looking at each of the endpoints and make sure that when you design it, you're going to be able to test your hypothesis and make conclusions based on your hypothesis, as opposed to just throwing some molecules into some animals to see if there's an effect. And you can always go back, if it's a small molecule, make a derivative. But if you've got a really good small molecule, you'd like it to be as clean as possible, at least at the dose levels where you'd intend to give to humans.
Yeah, I just had a
So, yeah.
follow-up question or comment
Sure.
on that. You know, it does seem like that would be advantageous. I mean, as you pointed out, because, you know, I think we've all seen, you know, like regular like grants and the like, where people are just throwing molecules at an animal without really understanding, you know, dose response or dose toxicity and those sorts of things. And so, you know, I mean, granted, I guess some people could figure that out later. That's a common quote I hear. But on the same hand, it seems like if you go about it in an informed strategy right from the get-go, it seems like you're probably more likely to target what you're doing in a reasonable and structured way, such that you're not like kind of wasting time and money.
Yep, that's exactly right. Even when you're designing your pharmacology disease models, keep in mind, if body weight loss is not the benefit you're looking for, it's a tox endpoint, record that, talk to whoever's going to be accountable for your toxicology, and evaluate that. I've seen that often where we're doing pharmacology studies and all of a sudden the animals are losing a ton of body weight, that IACUC has to step in and say, look, if you lose any more body weight, we have to take the study down. And again, that's going to stay with that molecule in that context for the rest of its life. So we want to be very careful about how we run these studies and how we design these studies. Another question here, is GLP toxicology mandatory by FDA or only non-GLP toxicology is enough? Yes, great question. To clarify, GLP toxicology is required worldwide for drug development. It's rare, and I give you some examples of when it's not required. It's because it can't be done that way. But yes, I would start off believing every first in human study is going to require GLP toxicology to start dosing humans. As I mentioned, cell therapy is a little bit different because we can't validate some of those tox endpoints because we can't use normal healthy animals like we can with small molecules and protein therapeutics, at least the majority of them. We have to use the hybrid model, which means that's an exception. Now, I will say, when you go to the FDA and say you're not going to do GLP studies, it is a visceral reaction. But it's become more boilerplate in the past couple of years, because now they understand it's like, okay, even though it's not a GLP tox study, we can't use normal healthy animals, do everything you can to justify and document the quality of those studies. So what we've decided to do is partner with Charles River, who has a very good reputation, in fact, a great reputation with the FDA. Like I said, we go to the same meetings, we participate on the same panels. I'll be a panel in Boston in a couple of weeks with Charles River. It's the World Congress, and the FDA's there. Peter Marks is there. So we talk to each other all the time to find what's the best way to do it. So even though you're not going to do it GLP, in your pre-IND response, if you ask that question, they're going to cite all the guidances. They're going to cite the law. It's the law that you have to do things by good laboratory practices. So if you're not going to go by the law, you have to justify it with science, with data, with rationale as to why you can't do it. And then the FDA has to agree to that. And they do. Let's see, another question. Can you speak to pharmacokinetics with biologics? If bioanalytics are less sensitive than ELISA, can you use ELISA? Yes, I have used ELISAs for nearly all of the monoclonal antibodies and bispecifics that I've got into the clinic, especially at Miramac, but even in general, I think ELISAs are most commonly used because I guess the main reason that over something like a mesoscale delivery type system is the availability of reagents, the ease of working with equipment and ELISAs. Of course, COVID had a huge impact on pipette tips and 96 well plates, everything else. But COVID aside, ELISAs are probably the main way that the main platform that I use to determine pharmacokinetics and anti-drug antibodies for protein therapeutics. Yeah, these are really great questions. Yes, it is harder to develop liquid chromatography mass spec for biologics, agreed. I haven't had much experience there. I always stuck with ELISAs and haven't had many issues, but there are times where LC-MS, MS can be more sensitive if you need lower concentrations or more specificity in your assays if the ELISAs are too challenging. All right, we finished therapeutic development. Let's talk about how it integrates. I'll I guess put in a little plug for myself. I presented this slide at Harvard and they said it was a beautiful slide. Granted, it was a friend of mine, but still it was Harvard. Got to take the prestige when it's given. This is just to summarize how toxicology exists. As I mentioned, it spans the whole drug development paradigm as early as you start using some evaluations for safety. Safety has to always come first, even in the discovery phase, through the development phase, and all through supporting your clinical trials. And then as I mentioned earlier, the post-marketing surveillance has a big part of it. Here's some examples of what types of toxicology happen at various stages. So there's screens in silico, in vitro, and in vivo that you do very early on. In silico, I mentioned the Derek nexus, where you're looking at the chemistry, the chemical structure, and trying to predict from that structure if you're going to have potential mutagenicity, carcinogenicity. I think the Derek nexus archive now includes some developmental and reproductive tox, as well as some general tox. So it'll try to predict liver tox or heart tox, cardiotox, neurotox. And then in vitro, if there's certain cell types, I know a lot of the induced pluripotent stem cell systems or embryonic stem cell systems have been evolved to attempt to predict cardiac tox, at least at the cellular level, or maybe if you get enough cardiomyocytes that beat in sync, you can try to predict if you're going to throw the rhythm off. That's a herd channel test as well. And then in vivo, if you do a quick single dose tox study up to that 1,000, 1,500 mg per kg, that could be non-GLP, a quick look in rodents to see if there is a toxic dose somewhere in there. And then you can move into the regular IND enabling paradigm, talk about that in a little bit, but this is just an example of what happens. And we've already talked about some of this stuff that happens during clinical trials. The regulatory stuff typically doesn't kick off until you get closer to your GLP toxicology studies and your GMP manufacturing. It's always a race between CMC and the GMP side of things and the toxicology GLP side to see when you're actually going to file your IND. So it's a nice little intramural competition within each of the companies. This is more detail about what you just saw, what's happening at the candidate selection stage. It doesn't give you the actual specifications, but it gives you the list of studies. I can provide this in a PDF form after this meeting if that's of interest to anyone. You can have these slides for reference, but you also have the guidances that can give you these lists. This is just a little more simplified form. Things you're looking for, right? Mutagenicity. If you have potential for mutagenicity, you also have the potential to cause cancer. So you want to be aware of that early on. You can screen rodents real quick to see if you have any of those vital organ effects. If you're affecting the liver, which is one of the main adverse effects, the cardiotox is second to that, and the brain tox, those main organ systems, or anything else for that matter. It's also captured in safety pharmacology, cytotoxicity, some of the other in vitro things, structure activity, relationship alerts, that's part of that Derek nexus approach. And then more recently, photoreactivity, UV activation of compounds, depending on the application, can be critical. Gene tox gets into the GLP space. You do some screening studies like an abbreviated AIM study where you're only using 2 of the usual GLP-5 species of Typhimurium staphylococcus. That's just a bacteria that you use that can predict mutagenicity. You do it more formally in a GLP environment here. Same thing here. You can do early safety farm screening studies. Maybe you go really high in doses and only use 1 or 2 animals. Whereas in the actual safety farm studies, you'll include more animals and have it up to a GLP standard. And then this is when you start getting into your 5, 7, 14, 28 day repeat dose studies in rodents and non-rodents that are required for your first in human studies. And we've already talked about some of this, so I won't spend much more time there.
I
Juvenile
wanted to
toxicity.
ask a
Oh,
quick,
yeah.
oh, sorry, I wanted to ask a quick question. You were talking about a couple of you might want to try this in those earlier screening, are there any like specific guidances that might be actually really helpful to some of the teams here? Like what might be some of those abbreviated testing pathways to kind of look for before they kind of get full-fledged into the all the GLP kind of requirements in terms of informing like some safety data without like, breaking the budget, so to speak.
Yeah, no, that's a great question, and certainly always a concern for any company. There are guidances that talk about safety studies, not so much focused on screening studies. Maybe Bill is more aware. Most of my experience with the screening has just come from what can we do cheaper, faster, quicker with smaller amounts of material and maybe not so many animals early in development. A lot of that has come from just working with CROs and being in the environment, the industry, getting feedback from the FDA. But most of the time, these decisions happen before you've even thought about discussing with the FDA. So it's typically going to CROs and asking them, is there something I can do that's cheaper, faster, more rapid turnaround, uses less material, uses fewer animals, or can be done in vitro to try to predict some of these other things. And that's something that I don't think is necessarily captured in guidances to a certain degree. Bill, what's been your experience there?
Well, my experience in trying to is kind of in the early discovery arena, which may be what we're sort of thinking about here, is that, a lot of people have developed in vitro models. A lot of them have looked at proteomics, genomics, and things like that. And while they can be helpful in discriminating between individual compounds, if you've got a large library you're trying to screen, they're not always that helpful and predictive in terms of the ultimate outcome. So, it's so Ken's right, is, trying to, I think, when you ultimately narrow down your molecules to the 1, 2, or 3 you want to move with, probably moving forward with some short-term in vivo type studies and around a mouse where you look at some of these types of toxicology endpoints is the most helpful. The one thing, genomics had a great promise about 10 years or so ago. And there was a lot of work done in the companies I worked with. But in the end, that data has turned out to be most useful when you're trying to address mechanism of action and trying to look at specific endpoints and things. It just was really onerous to try and look at every type of organ, and every type of change and genomic change you might have and translate that into ultimately an adverse outcome. Still a lot of work going on in that arena, but it's a challenge. I don't know if that's addressed what you want, but some of the dialogue around that.
Yeah, that sounds great. Thanks for sharing. There was another question. Does the FDA expect you to try LC-MS? No, the FDA just expects you to have a method that has been validated and can accurately, precisely, robustly, repeatedly, all the different things that you do in validation to generate data that you can rely on and predict what's going to happen in the future, which isn't always the case, but you do your best to assign the AUC and the Cmax and some of the PK parameters to the dose levels, and then you do allometric scaling or whatever magic you can to relate it to where you'd expect that AUC and Cmax to land in humans. And that's why phase 1 is always about safety and gathering PK data in humans as you dose escalate. So I don't think there's an expectation for you to use a specific method. The expectation is that you validate that method to comply with good laboratory practices, which is also defined in the law. How do we define MTD of large animals such as a pig? Maximum tolerated dose, I think, is common across any species. You just keep dosing up. It's kind of a dose escalation study where you dose until you see some kind of adverse event. Hopefully you don't see death, but any adverse event that makes the pig sick. Let's see, by weight loss yes, weight loss is probably one of the most sensitive and universal indicators of adverse events. If you're ill, you lose your appetite, you lose weight. So COVID-19 has an MTD as well. CBC, yep, all of these endpoints are well conserved across mammals. So you could use your complete blood counts, your hematology, your serum chemistry, clinical symptoms. All of these are tox endpoints, unless they're, unless weight loss is a benefit of your drug, right? If you're going for an obesity drug, then weight loss would still be a tox endpoint, but it's in this case beneficial, as long as you're not impacting other things. So yes, an MTD in a pig would be the same as defining an MTD in a non-human primate, a dog, a rodent. It may not be the same dose level, but it's the same strategy and the same protocol. So I'm not going to go through this. I know it's an eye chart. It's probably harder to see on computer monitors, but I just thought I would, again, I'll PDF this and it should be more visible if you want to look at it that way. This was just a small molecule IND Gantt chart that I created and thought it was relevant. Of course, you can see it's pretty old, but not much has changed, I guess is the point, right? I try to make things in red where these are the pivotal studies, the repeat dose, that 28-day IND enabling tox study can be a pivotal study contributing to the overall development timeline. But this was just to show you going from exploratory analysis, going through your DMPK early tox studies, so you can see some of the names of the studies here to give you a sense of what this particular program went through. And most of these are not necessarily required, but you may want to look at them as screening studies. So the hERG light is another way of looking at the potential for you to inhibit the human either agogo channel. Patch clamp is a better way to do it because you're actually clamping down on individual ion channels. The hERG is an ion channel, it's potassium ion channel. The Ames screens, again, typically you have 5 strains of the bacteria. In the early Ames, you might just use 2 to predict, etc. You can follow up with this, but overall, the point is it takes 22 months. It could be shorter, it could be longer, but you really have to plan the timing of all these studies, when you're going to have data, when you're going to be able to make a conclusion about your hypothesis, and when you want to make a decision to move forward with different molecules. It's a very regimented and structured timeline. Most people are trying to get it down to 12 months or less. And depending on your entity, maybe it's possible. Depending on your indication, you may be able to take more risk if you demonstrate the benefits, right? The breakthrough designation, once you're in the clinical trials, is a great relief because you don't have to wait that 1.5 years for the FDA to review it. They'll promise to review it in 6 months or less. You might even be able to use phase 2 data from a pivotal trial if you design it that way to conclude that it's effective enough to get on the market, and then follow up with a phase 3 pivotal trial after you've been selling your drug, just to try to increase the return on the investment so that you can help pay for some of the trials. This was just a flow chart, kind of a decision chart about what do you do in dose selection when it comes to the clinical studies. We've talked about the no observed adverse effect level. I have it expressed here as mg per kg. It could be mg per meter squared. There's conversion charts in FDA guidances. If you just search for estimating the safe starting dose in normal healthy humans, that will pull up the FDA guidance that talks about the allometric scaling and changing mg per kg to mg per meter squared within species, and then converting this to the human equivalent doses, which is the HED here. So I didn't define those acronyms on this slide, but if you looked up that guidance, it would have all this information. In either case, the minimum recommended safe starting dose is where you're looking at some of these factors. The human equivalent dose is divided by the safety factor. So it's just how to calculate what your starting dose is going to be in the clinic. So all of the work that you do in the pharmacology and the toxicology and in the discovery space contributes to one number, that no observed adverse effect level. So you're spending 5 years and millions of dollars to generate data to come up with that one number to say that it's safe to start in humans at this dose level. And we talked about this, that's the default safety factor is usually 10. In oncology, it's less than that. Sometimes it's 3 or less. And I've gone in with no safety factor because the benefit was so high, at least in the case of some drugs, that the patients don't have any other options. And the FDA recognizes that, and that's where orphan designation comes in. That's where all these specific types of designations, such as breakthrough and priority review and fast track, it's tried to stimulate innovation for those indications where patients have nothing to help them. And it's amazing to think that patients continue today to still have nothing for a lot of these indications. Let's see, how does the non-clinical timeline for an oncology candidate compare to non-oncology indications? Great question. I guess it depends on what part of the timeline you focus on. A lot of the discovery phase is very similar. Depending on the disease, a lot of the rodent disease models have a similar time frame, matter of weeks, between when you start dosing and when you see results. Same thing in oncology. It depends on how aggressive the tumor is in oncology, because it's hundreds of different diseases. So depending on what type of tumor you're trying to, it's pancreatic, ovarian versus head and neck cancer. Some are more aggressive than others, so you might be able to take the animals down, but it's still on the matter of weeks to get that data. The tox program is pretty much the same regardless of the indication. It has to do with the route and what your clinical plan is. You want to at least have a clinical synopsis that talks about the route, the duration, the frequency, and the dose levels to be tested, oftentimes informed by your pharmacology studies. But that 12 months from the time that you select a clinical candidate to the time that you file an IND is pretty consistent. Even for the cell therapy that we filed last year, we defined what the construct and the vector looked like. We had a time frame for manufacturing that construct and then introducing and transducing it into our cell therapy, which ultimately is termed a cellular gene therapy product by the FDA, which gets kind of confusing because now it's both a cell and a gene therapy. But nevertheless, the animal studies were pharmacology studies, so we didn't have to do the 28-day with the 28-day recovery and then wait 5 months for them to interpret all the histopathology. We kept it to the hybrid pharmacology toxicology study, and we were able to get that study done in about 3 months. So the timeline was still roughly 12 months, but maybe you can consider technically it was 9 months between when we locked down the final vector that was going to be transduced into the cell to when we actually filed the 2 INDs for both of those programs. Now, this is just another graphic to talk about how the therapeutic window can differ across indications and species. We've touched on that a few times in this presentation. In oncology, you can have a much smaller therapeutic window just because you can take more risk. These patients are, for example, metastatic pancreatic cancer patients typically have 6 months to live. So you're going to take a lot more risk in seeing just a little bit of benefit. When we got Onivide approved in 2015 through Miramac, that was only a 2.5, I think it was 2.4 month improvement in their life expectancy. So typically, if they're expected to live 6 months, they were going to live an extra 2.5 months. It doesn't sound like a lot of time, but it kind of wrenches your heart to think about if you personally have 6 months to live, 2 months can be a lifetime. You can do a lot in 2 months if you think that the next day you're going to die. And the other point is you don't want the therapies these people to go on, recognizing they're going to die eventually of cancer. You don't want the therapies to make them feel worse, right? Because if they're going to spend 2 months, their final 2 months with their families, I don't want them to be sitting in a chair hooked up to this device and worry about their hair falling out and all this other stuff. So that's why I was more enthusiastic to see some of the cell and gene therapies where you don't necessarily have to poison the patient. You can actually use the own patient's body to help regenerate itself and to help kill all the bad cells. That's what was more exciting to me with the cell therapy side of things. We still need a lot of small molecules though. This is a more elaborate description of what we talked about before with respect to the margin of safety. I won't spend much time here, but it introduces the concept of the MABEL, the minimally active biologic effect level. This came about because one of the questions that was asked earlier, sorry, it's late, about the effectiveness of a drug. There may be no way to determine the toxicity, or the toxicity or no AL is not sensitive enough. In the case of the T. Gennaro compound, this is where the MABEL concept came about, again from the UK, because the toxicity that we were demonstrating in non-human primates was not representative of the human population. And in fact, it was far less predictive and less sensitive than using what might have been an in vitro predictive mechanism of action approach. So I think we're evolving, continuing to evolve in toxicology and even in drug development in general to find better, more effective ways to predict safety and then dose escalate from there. So here we would use more in vitro data and whatever information we had from the pharmacology studies to say what we predict the mechanism should be in humans, instead of just relying on conserved historical values that would predict toxicity in a traditional sense. We combine the two, and whichever one is lower, right? That's the one we'd move forward with. This is just to have us on the same page of what an adverse effect is. Now I'm trying to be cognizant of time to go through these slides. Again, I'll send these with you so you can have them for reference. Why is something adverse or not? Usually it has to do with function. If something's affected at the histopathological level, like if we see necrosis, apoptosis in a liver slice, we want to look and see if liver enzymes were elevated at any time during the course of the study. So we look at the clinical pathology, we look at clinical observations. Did the animal look like it was functioning okay? Was there any kind of metabolic differences or challenges? So just looking at function of the organ that seems to be impacted, in addition to the morphology, can tell us whether something would be adverse or not. And then you have to figure out whether or not it's test article related. So these are the last few slides here, just to demonstrate in a 1-month tox study. This would be a typical study design. So put this table together. You've got males and females. You've got the dose levels at the top on the x-axis, the dose in mg per kg, 0.1, 1, and 10. So it's just a log scale. And then down the y-axis, you've got the tox endpoints that we typically measure in a traditional tox study. Mortality, did they live or die? Clinical observations, a few examples here. The body weight, we've talked about how that contributes. The clinical pathology, so this is where you're looking at your serum chemistry, right? And then the morphology, which is the anatomic pathology. And here it just lists the incidence of each. So in this case, you've got a control group. So this is just a vehicle control where they had some clinical observations, didn't have clinical chemistry for one reason or another, maybe it wasn't available. And as you move up in dose, you start to see differences in body weight. You start to see changes, whether it goes up or whether it goes down. You start to see whether it was reversible or not. That was another keyword that we put out there. And then if there were changes, there's a little check mark here. And then what you'll do with this data is here. You start to assign whether or not you think it's treatment related. And then for even the onesies and twosies that we talked about, you have to justify why is this treatment related and why something is not treatment related, why something is monitorable, why we don't think it's monitorable. That's an easier one to describe. Some things we can do ahead of time, right? Some things we can do in patients. We talked about the list of things we can see in humans and things that we can't see in humans. And then if something is monitorable and reversible at the histopathological level, because we can't take the tissues from humans. If we see something like myocardial necrosis, wow, we don't want to go anywhere near that, because we certainly don't want to impact the patient's cardiovascular system, something that's not as regenerative as the liver. So we'd have to make sure we assign an appropriate no observed adverse effect level to avoid getting anywhere near this in humans. In some cases, you can see there might be sex differences. So we're going to call that out too. Females might be more sensitive to certain adverse events than males. And we want to make that clear, and that gets carried all the way up into the clinical trials. And if it's relevant there, it gets carried into the actual approved drug label.
I have a quick question on the adverse events there. So, you know, like in the oncology world, how you would use like the coding system for human studies, when you're looking at animals, like, is there some kind of a quantitative tool? And I think the answer might be no, but is there a quantitative tool so that, you know, like across different drugs, and, you know, to kind of make it more standardized, you know, as opposed to, you know, like what some of those mean, like in terms of, you know, is it severe? Is it mild, moderate? You know, those sorts of classifications, because I think first I thought these were numbers, and then I realized they were the number of males or the number of females. And I just wondered, is there like any kind of system like that that helps to quantify that in some way?
Yeah, for the purposes of this presentation, I didn't go into that detail, but that's a great point, because incidence and severity are also a huge part of judging whether or not something is adverse or not. If you've got a minimal finding in your histopathology and it only happens in 1 or 2 animals, we can call it out, but we may not necessarily call that adverse, right? We might see some necrosis in the liver, but we don't necessarily, we think it might be test article related, but it's not adverse. So we'd still say it's a test article related finding. We want to track it in more chronic tox studies, and we want to certainly track it in humans. But the incidence and severity can contribute to whether or not you call something adverse or not. Yeah, it's a good point. I didn't capture it in this table for simplicity, but we always capture that in tox studies, and it is critical in informing the decision. Yeah. Another question, any disasters that you've experienced? Yes. If chemists or wherever you're receiving your material from have more aggressive excipients, sometimes those excipients can be the cause of toxicity and even mask your test article toxicity. One disaster I had about 3 years ago was we were formulating a molecule that wasn't easily solubilized. I think we use dimethylacetamide, which can be one of those more aggressive solubilizers, but it's also very toxic to rodents. And at the dose levels we were going, they needed 50% of it to be DMCA. So once we did the study, we found out really quick that even though it wasn't toxic to mice in the pharmacology studies at similar doses, the rats all died. And we had to make sure that we definitively said it was the vehicle and the excipient dimethylacetamide that caused the toxicity. So we even had to do a separate study to demonstrate that the test article in a different formulation with different solubility did not result in the death of all these rodents. And it's a hard battle, because like I said, once you generate the data, be very careful about the studies you do and everything that goes into that study. It was unfortunate that I came in after the fact that this happened, but I had to kind of help clean it up. So it did follow the compound up to a certain point, but once we got into the GLP tox studies and we had a formulation without any dimethylacetamide, everything was good, everything came up clean, and that was the data that trumped all the other stuff and reinforced the story that it was the excipient in those early screening non-GLP tox studies that resulted in the toxicity. This was just the same example going into dogs. You can look at this later on, and that brings us to the final part. Check that off. Yeah, well, there you go. This is because I'm getting to the Holy Grail. So I'm a fan of this particular movie, but you want to get the Holy Grail in that you can predict the efficacy and the safety, right? With something, in this particular case, it's just, I took this from some research just to show as an example, but is there a way for us to predict non-responders and responders from an efficacy standpoint, as well as from a safety standpoint? Is someone more likely to have adverse effects? Is there a marker, a biomarker that we can use to predict if they're going to have safety or not? And more importantly, are they going to have a benefit? So I thought I'd leave you in the conclusion with a question about the it depends, right? So is this a donut hole or is this a donut hole? And I'd say it depends. There's one more question here. But yeah, thanks for your time.
It depends what Homer says, right?
Yeah, or how hungry you are. There's one more question comment here. It's if one has a very potent small molecule that is working at single-digit nanogram per mil circulating concentration or less, and has a favorable toxicity level but is poorly detected by LC-MS, what is expected for PK measurements? Is it going too much higher doses where circulating levels are detectable considered an appropriate substitute when one can't reliably detect therapeutic levels? Yes, I mean, we default to using the dose as or dosage, the mgs per kg, or yes, in this case, might be lower than that, as a default. Even if we don't do pharmacokinetics, we're required to do what's called dose formulation analysis. And that's basically taking whatever concentration method your CMC group, your manufacturing group has, and transferring it to a GLP facility so that they can generation of the dose formulations that you're going to inject into the animals. So we can verify with an analysis that's included in your tox report that the concentration of the dosing solution was actually what was given. And depending on the route and the frequency duration, you can come up with further justification. That could be used in lieu of having PK measurements if in fact you're seeing biologic effects without being able to measure the compound in the serum or the plasma. Second to that, yeah, I would try to exhaust every possible method out there to find a way to measure your drug in the blood, because that is going to be an expectation. If you're early enough and you've done a lot of exploration, LC-MS just doesn't work and there's no other options, you can take the data to show the FDA or whatever regulatory agency, here's what we tried, and we just cannot get a method that's validatable to accurately or precisely detect the levels of our drug. Yes, that's a great option. Try dosing higher to get to circulating levels. If you can, if it's not too toxic, if you're seeing adverse events at higher dose levels, that could be challenging as well. Even if it's just single dose pharmacokinetics, where you give it one time at a very high dose and you can measure it for however long, hours to weeks, depending on the entity, that could be enough data to support going forward. But I would try to exhaust all methods and have that conversation with the FDA or whatever regulator you're going to, with the data showing, hey, we tried everything we could, and either they'll propose other options or they'll tell you what their expectations will be going forward. Yeah, good question. Great. Well, that was the end. Any other comments or questions? Oh, within a minute. Wow, that was really great. This has been a great presentation. Yeah, great interactions. Thanks for all the support, Bill, and the comments from Rich too.
Great.
And especially questions for Melanie.
Yeah, this has been a really great presentation. Thank you for answering, the questions, and as they come up, sometimes they're hard to follow if you wait too long to answer them. But yeah, thank you so much. I think this was highly informative in terms of, especially where most people are right now in the earlier stages about, kind of directions to head and some things that, you can do now in the earlier stages that may, have time savings later in terms of informing better your technology. So you don't languish while you're kind of trying to figure out what you're doing.
Understood and appreciated. Yeah. No, thanks for your attendance. Thanks for the questions. I'm glad it was helpful.
Yeah. I love the artwork in there too.
Oh, thanks. Yeah.
The humor.
That's more left brain stuff. Yeah. Have a good day. Good evening.
Great job, Ken. Thanks a lot.
Thank you, sir. Appreciate it.